I had introduced self-stretching to you yesterday, and people in my group I think have already heard a lot of what I'm going to speak now in bits and pieces. So let's try to see the overall picture, and I'm showing those things on the slide. Please ask me questions if you don't understand, or if you have other questions I will sort of try to address. So this is something that basically has not been my real area of expertise for long. This was sort of something that we needed to do for understanding what we were doing, and so that's why we got interested in doing it. And so why stretch cells? Cells, there are various kinds of cells which undergo a lot of stress in their lifetime, or a lot of you are from biomechanics background, so you, I'm sure, already conversant with that and would like to study these cells under mechanically stressed or stretched conditions. So the resting state is very different from the in-stretched state, and however, so we are talking about smaller length scales, time scales. If you cells adapt when you take them from the resting to the stretched state, and if you keep them for long in the stress conditions, so finally, after a chronic stress of stretch of a day or two, it would sort of get back to a new chemical biological state, which is no more stretched. But there are other kinds of systems where we go through continuous cyclic stretching all the time. So that's a completely different kind of stretch. And so these are the different scenarios in which you would like to study your cells, which we usually study in a completely resting kind of state when we do in vitro cell culture assays and look at different proteins or factors of the cell. So what is really remodeled when you do a stretch? The first thing that people have seen is that, again, we are trying all the time to compare from 2D cell culture assays to 2D cell culture assays on stress substrates, and to think of how the cell responds. One response of cells is cells reorient themselves with respect to their cell shape, depending on the direction of the stretch, the frequency of the stretch, and the amplitude. The actin cytoskeleton is remodeled. There are signaling events, so this is a completely different, one kind of problems that are addressed usually. I am mainly interested because I want to understand the strain state of the membrane, and we want to understand how, when you strain a cell, how much does the membrane really get strained, and how does the cell regulate this response of the cell in smaller and longer time scales. Okay, so I talk about, again, thinking between usual growing cells on a petri dish or a glass surface, which is not stretchable, which is rigid, to growing cells on something that is stretchable, which is rubbery, like you have a elastic rubber band. And so this nice polymer is called PDMS, polydimethylsiloxane. There are other kinds of polymers out there, but this has come up. This has come up because this is one of the most biocompatible, and there are other things which will take you through, which are good about it. So what is it? This is how the polymer looks, the chemical formula of the polymer. This is the mur, which we are basically polymerizing. The three polymer are short chains of these. You add a cross-linking agent, which would take many of these small polymers, cross-link, so you have another cross-linker, so it now becomes a mesh. Now the fluidic properties of this three polymer changes when you add the cross-linker and you let the cross-linking reaction. People in my group, we did that yesterday. When we added the cross-linker, you can leave it for a whole day to cross-link, or you can fasten the process by keeping it in a oven at 60 to 80 degrees centigrade or higher, and do it really fast. You can help the reaction finish in about an hour. Now what forms, we handled this thing and we saw that it was really like honey. It was more viscous than elastic. But as Pramod yesterday told you, that these kind of polymers, you can always have, you can measure the viscoelasticity, both the viscous and the elastic properties. After the cross-linking reaction, what happens is this becomes more elastic, which is good for us. We can handle it, we can stretch it. And however, that once this cross-linking has been done, what you have in hand has certain properties which some of them are good, some of them are not so good, and which you would like to tune. And so let's just take an overview of that. Okay. A lot is coming up, and of course the field is changing, but this is to just give you an overview. So the first thing that is very sort of drastic is that it's very hydrophobic. So if you put, and that's what you would not like it to be, because you are now wanting it to go into water, water-based media, and you want cells to spread on it and the water to interact with it. So what you really have is that a very high contact angle, so the water would just sit like a drop and would not want to spread at all. But good thing is that again you can do surface chemistry and change the hydrophobicity there. Now, so this is bad, but you can make it good. The diffractive index. So the diffractive index of this, again, this substrate is around 1.4. You can change it to from 1.4 to I think 1.434445 by baking it. I have a plot in the from a paper, and then I don't remember the numbers very well. So again, so it's not so good because glass is 1.5 and most of our microscopes are adapted to work with glass. So 1.5 is a nice number, but then we also work with water immersion objectives and so on. So it's not so bad because at least it's more than the refractive index of the cell inside, which is approximately 1.3 to 1.38. So you can still have a higher refractive index media through which you are now wanting to image. Okay. So again, right now we are talking about this because we are talking about imaging through PDMS. Of course, again, because of the bias of what we do, I'm here presenting again it as a substrate where you grow cells and stretch. But it has other applications. For example, you have contact lenses which are made of PDMS. So for a completely different reason, not per se imaging, but it is good that the refractive index is something which is more than water, so you can actually use it as a lens. So that is one thing. And with respect to surface chemistry, it's tunable. So I've just written one word because you can do a lot to it. You can oxidize it so it becomes more like glass, and then you can plasma treat it. There are more free radicals on it to which you can add things. You can do covalent chemistry. There's a huge lot of things you can do on that. Okay. So now the other thing is that, as I said, when you go from a fluid to you make it elastic, you can make it elastic of different stiffness. So it can be a soft elastic, it can be very hard, and so on. But how bad is that? That's a good thing that it is elastic and we can actually stretch it. If it were like glass, then you wouldn't have been able to stretch it. So these are certain, and this is a very recent paper in 2012. This was a question that came up when we were doing our yesterday's workshop, which is that what is the range of stiffness that you can take PDMS through, and how good or bad is it? I think there are, of course, there are other ways to change stiffness by growing, like I think Firoz might talk about it, I don't know if he's not about it, by growing patterns on it or having PA gel which can take it to a lower. But this is sort of a commercial pre-polymer, and it has their own patented cross-linker which you can use to make PDMS. That's what we were using for the workshop. But there are other kinds with modifications which I really don't know about, and you can see here that mixing these two kinds of pre-polymers, you can go from something like really low, so I think it might be 10 kilopascals here, from 10 kilopascals to almost 2 megapascals. Okay, so you can have a huge range of stiffness with these substrates. It's not so important just for the stretching, because when you're stretching, it's okay. If you need to give it more force to stretch, you can give more force to stretch. What you're really interested in there is this strain, and that's fine. Just for using something like which is whose stiffness is here to here, all you have to do to get the same strain is apply different forces. Where this becomes important is when you are also worried about how cells which like certain kinds of stiffnesses. When you work with cells, different kinds of cells which like different kinds of matrix rigidity. Okay, that's where it becomes more important, and that's why it's up there. Different ways of doing it is the cross-linker that I said. How much of the cross-linker you use can change it. So I think which is this. You can go from 2 weight% to this. Yesterday we used something like here, and by just changing that, you can go almost 1 order of magnitude in the stiffness. And if that's not good enough, then you do that, and you can also play around with that. Okay. One thing to remark here, which I may not come back to, is that that you can assign a stiffness to this polymer only works when you are working in ranges which is less than 10% of stretch. And if you remember when we worked yesterday, we like 20, 30, 50, almost like 80, we crossed 80. So, and then we discussed if the stiffness actually changes with doing this, and Dyke's talk yesterday in a different context showed that if you stretch something because of whatever various kinds of reasons, it can strain, it can change. So it's good to remember that about PDMS, that these numbers are for numbers which are below 10% stretch. So that's the linear regime after which it becomes non-linear. It becomes more difficult to strain it. Okay, it becomes more rigid, and of course, remember it's important. Remember that after a point it will break. But a good thing is that that's a big enough number. It's not. I mean, it's not really as good as even rubber band. Rubber band can take around, I think, 15 megapascals here, or human skin is around 30. But it's good enough for our experiments that we have seen yesterday, doing almost like 100% stretch, and it doesn't break. That's fine. So yeah, so this is sort of linear only in this regime between a 10 to 20, 0 to 20% stretch, after which it starts becoming non-linear. And so there, I mean, what you have to remember is that you cannot really use the numbers you are using to strain it. You cannot say, I applied a 10% strain, so my cells are 10% strain. It can be non-uniform even over the PDMs that you are using once you cross this 10% limit. Yeah, so therefore, while it is good, it has its caveats, and these are the caveats that you have to take care. So the not so good is that it is optically transparent, but there's so much more to say about being optically transparent, and this is what you see here. Glass. This is the transmittance of the material, and this is over the wavelength. So it's good that is most of the wavelengths that we use is pretty much transparent. But how transparent it is, it's around 85%, and maybe with better chemistry, better clean rooms, this and that, you can sort of increase it, but you cannot take it as good as glass, which is more than 95% transparent. Okay, and this is also a limitation because the kind of polymers we use, they usually have nanoscatterers which are hard to get rid of, and that does affect the kind of that creates scattering, and you have this kind of transmittance. However, one thing to remember here is that if you use thinner and thinner PDMS sheets, this problem becomes less and less problematic because you have less and less number of scatterers that your light path is going to. Okay, that's another good thing about. Besides, I think that yesterday I talked about why we need thin PDMS sheets is because we need to reach out towards the cell with an objective which has a high NA. But besides that, this is another reason why you need to if you make thinner and thinner PDMS, it's better for you because your transmittance gets better. Okay, I talked about refractive index, and we come back here where.
Sorry, Bidisha, how thin can you make it? Because at some point in here,
yeah. So
and that will depend on the percentage weight of the polymer.
Yes. Yeah. So if you can work with, so again, if in your application if you can work with 100 micron thin PDMS, you can easily stretch it to 50%, 60%. I don't know how if you can work with 50 micron.
Yeah. No, I just think even just as a support for growing cells.
Support on glass.
Yeah. Oh, so this is on top? No, this is on glass.
No, I'm talking about unsupported.
Right, unsupported. Right. So
you can stretch it.
No, without stretching. Is it very what do you call it? I suppose you can't have a very thin sheet of PDMS
because you can't handle.
You can't handle.
Yeah, with air, with air, the objectives it's not the problem. With 150, I have not. 50 even handling it to put on such is a problem. 100 is not a problem. 100, 150 is not a problem. Below that, if you go and if you can handle it, of course your optical properties will increase. Yeah. But handling becomes easy. Yeah. But
this is at what? This is at 100?
This is, I have to check. This is much more than 100. This is much more. So we have not characterized it with, I think that's one thing that one can do. Taking glasses, using glasses transmittance, use a different thickness of PDMs and see what the difference is in the transmission. Yeah, so
it's pretty bad.
it is bad.
Yeah.
It is bad.
I
And
don't
this
know
is
how
for,
thick this
I have to take a could be around an mm. So, yeah, this is pretty bad. But I
because because if it's also scattering, you lose resolution.
use those resolution. So even with a 100 micron,
Yeah.
where you have to, I'll show you certain images where you have to see 50 nanometer objects or 60 nanometer objects, you would need some amount of averaging. Take out the noise, but you will still not see as sharply. But another test which failed was using optical trap theory because the scattering profile is really bad and you cannot. So here is where it's important for, this is something that we are really dependent on the chemistry to develop. So where you can take off.
So is it because it's incomplete polymerized material?
It's because during polymerization, the material forms aggregates. That's yeah. So how you? What? These are defects. So basically, even with the various kinds of baking techniques, whether you use slow or fast or gradient, people still cannot get rid of this. But one thing that can help is, which is not very well quantified, is aging. If you let it age, if you let the PDMs age, then a lot of these properties go and I don't know why that happens. But at least in refractive index of the material, you can see that it goes 0.02 in 1.45. But so change in refractive index, again you lose light when you have a refractive index change, and this is just 60 minutes, and I keep it overnight, hoping that it will reach whatever it can reach. So to find the paper in the morning, I didn't find it at the laptop, but there are other measurements people have gone to 1.48 with longer and longer and more high density. So again, this I think it's the chemistry which has and also the kind of polymers we use or so on are not very well are patented. So not that I would be able to dig up why is it happening, but you don't know the chemistry at all. So So next is the various ways of stretching. Again, people from my group yesterday, what we did was the simplest way of stretching. You hold one end and pull the other, and the thing stretches, right? So that's the uniaxial. So I have not drawn it because we'll be talking about it again. The biaxial. The biaxial is tricky because you need to stretch it from both directions. So how do you do it? Do you just clamp it in both directions and stretch? Then you can have edges which can tear. This is a nice way that people have done. People use this as basically how you do it, and this is another modification, 98, 2010. This looks better, and you can do better imaging through it. Here, basically, you have a membrane which is on a circular surface, which is on a ring or a tube, and you have another ring, which is the O-ring, on which PDMS can slide. Okay, so you put a sheet of PDMS. You come with something which can screw on it, to which you can attach the PDMS. So not that you're screwing it, you're not screwing it. So that is also, there are no threads there. It's all free here, through. So what you can do is you can move it with a linear actuator or whatever. And as you move it, the whole of the PDMS which is stuck to the ring is actually, so you're converting a linear motion to biaxial stretching in this direction. Okay. So you see that this is a more sort of a complicated design that we did. But if you're really interested in biaxial stretching and uniaxial stretching is not going to tell you what you want to understand about your cells, then that's the way to go. Now here, this design of course has problems because it is a fixed plate here and so on, so it is not really transparent. You cannot use the here. But this is a more sort of sophisticated design where you can do it, but it still makes the access to the membrane a problem. Okay, the whole design, unless you can even in this design, I think the best kind of objectives they could use was 40x air objectives. So. But I'm sure, one can work around these designs to make them more amenable to your experiment. Now, okay, so this is one way where again you are basically 2D. In 2D, you are increasing your area. The uniaxial that we yesterday talked about and showed in the workshop was again 2D, and you're increasing the area. The third way to do it is to forget about 2D, to have something like this. You have a membrane here, and you can really push on the membrane, okay, with air or water or whatever. You have a pressure difference, you push on it, and of course this thing is like inflating a balloon, so the area changes. So here you are not retaining the 2D geometry, but you are stretching yourselves in a sort of 3D way. And this is more, I would say, in many contexts, for certain cells, this is the physiological way of being stretched. Okay. Now, how you can couple this to imaging, it really depends on what kind of imaging do you want to do. If you want to keep in contact with the cell, like oil immersion, and do it, then it's not good. But if you can come with something which is like the air objective and still have good images of the kind of structures you want to see, then it's good enough. And you can do a 3D scan and again get the shape of the stretch cell. Yeah. So this is our stretcher for those who were not there. Very, very simple design, where this is a piece which is always on your microscope. Okay. So before we go through these, let's look at the key requirements of why we have a stretcher like this. And this is only for a particular set of experiments. I mean, the whole idea is to take you through one of them, and also a very simple one, so that you can go back and have a different one which has different key requirements. One key is it has to be compatible. For my experiment, it needed to be compatible with the inverted microscope, small working distance objectives. Okay, so therefore I needed the membrane to be right at the bottom, and nothing around it which is blocking the objective from coming close to it. Okay? In X, Y, Z where. So the membrane actually goes down. So maybe I can draw it there. Okay, and I want to image live cells during stretch. The other biaxial stretching, so you might say, then why was it useful here? Why did people do it? People did a lot of work on these, because what they did, they did not need to look at the same cell in the live state during stretch or just before and after stretch. They could work with an ensemble of cells, not stretched, stressed, and still get a lot of information, right? And that will do. If you have something like this, you can fix the cells after this percentage of stretch, whatever frequency. You can change the frequency, amplitude if you want to play around with cyclic stretch, and then you look at either you take the membrane completely out and image the cells with any kind of objective you want, or do biochemical assays, right? So here, why we are stuck with the small and little design is because we want to image live cells. So we want the cells to be alive when they are on the microscope, and we want to monitor them continuously while we stretch. And the other requirement was, because we do single cell experiment at a time. At one point in a course of an hour, I look through one cell. Okay, that's not a lot of experiment. And if you want to get more statistics, you don't want to keep the whole thing in the incubator, grow the cells, and then bring it and then do one cell experiment in 2 days. You can't do that. So you need something to be removable, just like putting a petri dish on your microscope and doing an experiment. So it needed to be modular, simple design, and multiple samples should be loadable easily. So this is the design where this part just comes out. It's as good as a petri dish. It can go to your incubator, and whenever you need to do an experiment, you can bring it back and just attach it to the motor and start your experiment. Okay? That's the design. And let's see the design a bit more. This part is completely rigid. This is all connected. There's nothing mobile. This is the only part which is mobile. Okay, and between this and this is where you stick the PDMS on the bottom of it, not on the top. On the bottom is completely flat, and that's where you stick your PDMS. And when you move this part, naturally one part is kept fixed, the other part moves, and whatever is inside basically is uniaxially stretched. Now, this slit here is basically to allow you to have a screw here, so what you can do is what you need to do usually is you need to put the PDMS, which you do with your hand, and people have done it to know that it's really tricky, it's very, very sort of floppy, so you need to handle it well, and even if you handle it well, you will have certain kind of undulation which you want to remove before you put your cells. So you want to pre-stretch it. So if you're pre-stretching it, naturally if you leave it, it will come back. So you want to pre-stretch it and lock it. at a particular stretch. So with this you can do that. You pre-stretch it, lock it at a particular stretch. You grow your cells in it. We can talk about that in a while. And then that's when you put it in the incubator in a moisture box, which is inside the incubator, which stops evaporation. Let us talk about the length scale a bit here. This is actually 1 centimeter. This is therefore around 4 or 5 centimeters, no, or less. So you start with something which is 0.9 centimeter long, 0.5 centimeter wide. You pre-stretch it so that it becomes 1 centimeter long. So we're already in the edge of the linear regime, but I'll tell you why you don't care too much. Because the kind of pre-stretch you really need to remove the undulations. It's basically when you pre-stretch it, you're not stretching it. You're just, in fact, I correct myself. I shouldn't say you are in the out of the linear regime because you're not even started stretching. But you don't know to where you stop because you do slightly stretch. So you start at something like 1, because this guy is big and there is some space here. Basically, you can do 100% stretch in this. Okay? You can go from 0 to 100% stretch in this stretch. Okay. Now, as I said, you can put it at any point and lock it. And in that locked stretch condition, you can put it in the incubator, which is the pre-stretch conditions. If you want to use it, I don't know, for some reason, if you want to give it a big stretch and then look at, put it in the incubator, or look at it, put it in the incubator, come back and re-stretch it. Those things you can do because you can lock it, and whenever you want to work with the on the microscope, you come here, you first lock it to the motor, then you unlock it from here and let the motor do the stretching. Okay, that's the modular and simple design which we have come up with here. This of course becomes a bit bulky. It's not so bulky, but still, if you just want to use it on the microscope, it's fine. If you want to put a 37-degree incubator on it, the on-stage incubator, there's not much space around it. So, but we still want the thing to be heated. So again, I told you about dimensions, and let me just tell you, if I can find a marker, as to how we grow the cells. So this is a PDMS sheet. So we take silicon gel or silicon grease or whatever you can call it. We have a syringe in which we fill it, and you sort of press hard and you get a tube, sort of a tube of the silicon grease, which we draw here. So you can draw it properly, or you can draw it whatever. So you have a sort of a silicon grease which is sticky to the PDMS, right on the PDMS, which is where you can put your cells. And what is the kind of volumes we are talking about? From 50 microliter to 100 microliter. These are the volumes which that little thing can take. Okay. Now you might wonder, why can't I sort of increase it? This is too difficult to work. Again, the question that Jiku had also asked was, how much can you handle it? We are working with 100 micron thin PDMS sheets. If we go beyond this, in fact, yesterday we tried an experiment with the cells were nice. So usually we work 0.5 and 1. If we try to make something which is 1 and 1 or 0.8 and 1, it starts to become more strong. But if you can handle it, fine. But as soon as you have to remember that when you put the silicone grease, it again creates a sort of a non-uniform pressure on it. So all these things one has to keep in mind. What we have come up with in the lab is that it be like this. This is the aspect ratio we like, and that's why we are stuck with something like 50 to 100 microliter volume of a liquid. What we have to really worry about here is evaporation. So in the incubator, what we have is a moisture box. So there's a lot of water here. There are some holes in which your stretches can sit, and we sort of slightly close the lid. There's a good amount of, it's as humid as outside. Maybe it can basically, it's a smaller place where your initial evaporation is even less than if you put it in straight away in the 37-degree incubator. So the evaporation here is, we have not really quantified it, but overnight you do not lose your 50 microliter of fluid. I think if you just keep it 50 microliter fluid outside, you definitely dry. You don't dry it. But over the time scale of the experiment, our experimental time scales are 1 hour, so therefore the loss is even less. Though you do not have, actually not less, it could be more because you don't have the humid environment. So what you can do there is we put up a little drop of mineral oil which can go and cover the top and stop evaporation. For a matter of hour, it should be fine. I don't think the oil would sort of start interacting with the membrane or anything, but this part really is I would say sort of tricky. So here what I wanted to say was, since as I said we cannot really put it in a 37-degree incubator, you can have sort of steps here where you can put thermistors and you can heat the thing up with just putting voltage of the thermistors here. So this can get heated up. This is all metallic, so the whole thing can get heated up. Yeah.
I was just wondering, what kind of cellular densities do you work? And since you are plating them overnight in a 50 to 100 microliter volume, and there could be possible concerns from nutrient deprivation and things like that, because it's a very small.
Okay, so here let's talk. That's a good point. So let's talk about how to prepare the sample. Okay. So as I said, PDMS is really hydrophobic. So you cannot really just put the cells. You do something about it. So let's talk about the simplest thing which we are doing here, which is to give it a plasma cleaning treatment. So you have free radicals and then dump fibronectin, so that you have approximately 2 to 5 microgram per centimeter square of fibronectin sticking on the PDMS. We can put cells in this, and the density I'm talking about is, I will not tell you per ml swimming cells, but I can tell you that the approx, so these cells are around 20-30 microns, and we calculate numbers so that they are around 30 microns away from each other. Of course, in certain cases we have certain clumps and so on. Important point is we do not wait overnight, usually, unless there is problem with sticking, do not wait overnight to do the experiment. If you have fibronectin and if you put cells in this density in which you would like to work, in an hour they spread out. So, example, yesterday, before Pramod's talk to, before dinner to after dinner, and we had a short dinner, they had nicely spread out. Whereas in the same time scale in glass, without fibronectin, they were still rounded up, and PDMS with polylysine, they were still rounded up. So, if you have fibronectin, you can fasten this process of addition and spreading, and you do your experiment right now. So, you plate the cells in 1 hour, they are ready to be.
I have 3 questions. First is that, does your media see the silicon gel?
Yes, it does.
So, does not that silicon
It's
gel
not
bleach?
supposed to be toxic.
It doesn't bleach anything?
It should not. It should not. Both PDMS and silicon gel should not.
PDMS is a cross-linked, so I don't expect it to bleach anything.
Oh, you mean it doesn't come to the solution? No.
And second, you are coating your PDMS before the stretching. You are coating your PDMS with fibronectin before the stretching.
Yes.
So if you have a different level of stretching,
You will have. So what you will have is you have surface which is unmodified exposed, right? But you are not going to plate cells anymore. So your cells are in the sort of have the surface.
But I mean, my question is that if I want to compare between 50% stretch and 80% stretch, my
Be a bad number when you do it.
my linear density will be different, right?
Linear density will be different. Density per se will be different, but your receptor density is also different. So ratio between ligand and receptor is same. Of course, it will not saying that there is more receptors being transported to the membrane. So fibronectin density, of course, is changing when you stretch. And since you have, maybe we can figure your third question and come back to your second question is a longer answer. Yeah.
My third question is that when you plasma oxidize PDMS, it creates a hard.
Right.
So when you
Right.
stretch
So there is where you have to play with the time series. If you do like a nice 30-second plasma treatment, you have sort of a layer of glass. I was saying, if you stretch, you break it. You have breakage. So you have the surface cracks. So you have to do just sort of 5 to 10 minutes. Okay. You don't let it really form a whole layer, connected layer. You have certain points. So if you are hoping that only certain far away points are getting oxidized. And it's not so continuous that they can like form something which cracks. So you have to play around the time 5 to 10 seconds and all. So that's a very good point. And again, this is something that we can use. Maybe at one point we thought we can use the cracks as patterns. Patterns you can, of course, you can do micropatterning, but in the absence of it, you can use this to create patterns. So again, to talk about the ligand concentration, there's another thing you can play with to have a surface which has a gradient of ligand concentration. And so here is where all the markers are more.
Remember?
Yeah, this is better. So now if you have, so I forgot to put that, but anyway, if you have a rectangular piece of PDMS and you are stretching it, so of course it's not stretching like this. Where is the, so it would sort of stretch like this. So you have a contraction in this and an extension there. On the same person is equal. But this, of course, that depends on how much that will, how much of this contracts, how much of that depends on the material properties and the Poisson ratio. Now, if you have something like this is something we are doing in the lab to exploit this. And now if you stretch it, this points here will get more easily stretched than points here because it has sort of support. Now, one thing you can do here is you can use this to have non-uniform strain. Fine. So if you want to assay something, what we are doing in the lab is trying to understand wound healing and the effect of strain on wound healing. You can ask me why. But here you can have, for example, a wound which spans this. So in the same experiment, you have the effect of strain. Strain has a gradient under it. Now, coming back to the ligand density, you can do your fibronectin coating on whatever ligand you want to coat and then strain it. So because the strain is non-uniform, the ligand to ligand spacing will now be non-uniform. So you have a gradient of ligand. Then you can put your cells. That, and you need not set it. If you just want to create gradients, you can actually use this whole concept. You can strain, have a gradient, and pull it back if you want to have the other reverse. So these are other things that you can do.
So why did you strain this? How did it transfer to the ligand? Did it any person with this, or is it more sufficient?
Okay, what we're talking about, I don't know, the ligand, we're not talking about straining the ligand per se. All we're talking is that if this is one ligand, this is another ligand. Okay? This is before stretch, and after stretch, since the thing has become bigger, so let's say we have done it like that. This is here, this is here. So concentration, what you mean by concentration is that how many ligands are there per millimeter square. Okay? So which is less now. That's all we're talking about. We're not talking about stretching or straining the ligand. Okay. I hope I am okay. I don't have too much time. So again, so this is what I showed yesterday, and it's fine. We have sort of covered it. You can either micropattern or just have cells get the right kind of density. And because we have thin PDMS, we could even push it and try to do turf. And these are images where caveolin 1 GFP is expressed by the cell. They form small 5200 nanometer dotted structures, not claiming that each of them are 5200 nanometer, they can be clusters. But you can sort of start resolving it. But it's not something that you can do in 100, 300, with glass. You can take a 300 millisecond in the same kind of camera and just giving estimate, and you can have an image like this. With PDMS, you might have to do a 3-second averaging. It really depends on how your cells are sticking, how your PDMS. So that's why you need clean room facilities usually. When you make PDMS, you're going from a non-crossing or a fluidic thing to crossing things. Okay, and this has been completely exposed. Yesterday you saw we did that, and then we realized that when you mix the two, you have to mix really hard because both of them are viscous. We realized that it was important on what we are mixing it in. This was nice, clean perhaps, but we used, and maybe the environment was also clean, but we used plastic cups and we did that, and we think we scraped plastic off. Now this is not going to remain on the surface. With glass, it's good that you can clean it at any point you want. The rest of the surface was not in your hands, the company needed to do it. Here you're making it, so whatever dust or plastic or whatever you introduce is going throughout the PDMS and making it dirty, and your optical properties are basically being hampered. So what we yesterday did was really a hand-on thing, which was give you an sort of. But when you do it for an experiment, you have to be clean, and that's why we So now, yeah, if you want to do, it depends on what kind of imaging you want. If you want to do something like this, it's advisable to have cleaner environments. You don't want to polymerize dust with, okay. This is from a paper whose reference is, I forgot to put the reference there, is this one, which is cool. I think really nice if you want to do, if you're happy with this kind of imaging. This is that in situ skeleton, and okay, when you just take a look at that, it doesn't look that the cell is stretched, right? I don't think it's stretched. We want to see something really stretched, and that is, I can tell you, much beyond the 10 or 20% strain that we're talking about. This is 14% strain, and you would hardly be comfortable calling it strain. But no, what you can do. So what I told you, that if you just take perimeter strain, you can go to nonlinear. It might not nonlinear regimes. It might not be uniform throughout the sample, that 1 centimeter pointer, but you don't care. What you really care about, if you are doing one single cell experiments, if you're doing many cell experiments where you want to have biochemical assays, then you have to really care if the whole PDMS is uniformly stretched or not. Okay? But if you're doing single cell experiments, then you will have to basically go back and compare the image before and after stretch and get something like this, where they have, you have the image registered. Basically, you have patterns recognized here and here. Compare them and find how much the thing has stretched. What we do is, we used to do is that it's known that if you stretch cells, then cells start to reorient. They don't like to be strained in this direction. They will start remove their focal additions, form new ones, and get reorient. They can become completely perpendicular to the direction of stretch. It depends on how you do it. They can be parallel to the direction of stretch, and so on. So what we do is we already do not allow that. We do not give excess area for addition. So there is no scope of rotation. So you have a rectangular patch, and that's it. The cell has to sit there, and when you stretch, you just see how much the cell is strained in that direction, and that is your strain. Okay, so you can do both. You can let it. So this is really not micro pattern, but the cell was shaped like that. So I got that. You can let it stick as it wants to and do an analysis like this, or you can decide how you want it to stick and strain it, and then just take the number. These are different ways you can do it. Okay. Continuous scanning, I think we talked about it, but let's say these are the 2 points. Okay. Let's say this is your stretcher, and this is you need a motorized stage if you want to continuously see being stretched, and this is why. Let's say these are the 2 cells, and this is the cell you want to study. Okay, this is where let's say objective is. They were here. Now you stretch, and you stretch in one direction. It doesn't matter. Even if you were stretching in 2 directions, the only point which will not move from your point of view is the band center. And you will not always come to that. You will like a cell which is nicely fluorescent, nice shape, which is in a completely different position, not at the band center, right? So if you do that, then let's say it's at one time point it's here. When you stretch, this moves, that moves. This is just done hand-wavingly. So in the stretcher will move, that's why this and that move. So you have moved from your visualization. And it can be, if you're looking at bigger areas, you have bigger strains. I mean, even 10% strain on 1 centimeter, you have 1 millimeter movement. So something which was, let's say, almost at the center might move 0.5 mm. Okay, 0.5 mm is a lot for your field of view. The field of view is 100 micron, not more than that. So while you're doing the stretching, you have to keep bringing it back to the center. Okay, so this is how you do it. If it moves, you move the whole stage back. Okay, and this, therefore, you have to either, if you're very confident of the velocity of your motor and that you can start the movement of this stage and this stage together, then you can set the velocities depending on where you are with respect to this edge, so that both of them are moving and you get that. But what you will still see is that cells moving away from each other. The other way to do it is you do a shot before you arrive at the 10%. Let's say you do it in 0.1% steps. 0.1%, take images before, after, see how much it has moved, and move back your state. There are 2 different ways you can do it. I'll show you if this movie works. Okay, this was to show you that we can do turf, but there was some problem with the movies. Let me show you 2 movies. Let me show you a nice movie first. Okay, these are myotubes that bar is I think 100 microns, so this is huge, or no, that's 50 micron. And you saw that so don't worry about what they're loaded with, that's just to mark the volume. So you see that it can stretch like that, and you see this cell, suddenly the intensity changes. So this cell has basically burst, okay? And the other cells are not bursting. And such events of bursting are really, really rare. So let's see that again, and you have a view. So you see that guy is moving much more than these guys, because it is at a different position with respect to the edge. And this is not high resolution imaging. This is 10x or 5x, because the cells are huge and you want to see the whole cell. But this is how it would look, and this is how it was done if you did not completely center it properly. Okay? This is what I mean, that things move away from each other. Here. So the center is moving, the whole thing is basically moving. But the good point is here, what you have to see the good in everything, is that even though you're looking at 10x and you're moving a lot, this thing is more or less staying at focus, which is not bad at all. For something floppy, done machine, not to very much precision, when you stress like this that you're not moving too much in Z is good. Okay. So yeah, so this is I think what I yesterday we had problems of the PDMS being not very flat, so we had oil problem, and we had problems with the, so we can, if we list the problems, we had more of them, I think. We had the software deciding to forget about the blue filter and just blast the whole spectra of the lamp on the cells and finally lost the fluorescence. So today we are going to do just imaging. We have a lot of samples and yeah. So yes, first day our cells were not growing. Second cells grew when this happened. So yeah, the last thing I want to tell you is this is, so in case so right now we have GFP, GPI marked, and that's what we are trying to image in quantifiable. But our backup plan, in case our fluorescence we can't really catch it in quantifiable, is to really do this, where you are loading the cells with something which is fluorescent and cannot escape from the cell. It's catching air. And it can escape only if you rupture this. Okay. But to give you an idea, and I really like it. Oh. It's a very sort of bad experiment that I can't try it on you guys. Is these were our blood cells, red blood cells that we just somehow put on we had sort of a polylysine kind of thing so that they stick on the membrane, and we stressed, and we could see multiple cases of rupture. It's hard to make out rupture, but it's a different way in red blood cells to make out rupture. This is a fluorescent lipid on the red blood cell. Even on a ruptured one, you see that. But what you lose is on the bright field image. So this is a cell which is ruptured. It cannot be seen in bright field because there is no change in refractive index outside and inside. So you can't just see it. And we had to do this. We could not follow the cells while we were stretching, but we could see in the whole sample lot of rupturing events. With eukaryotic cells, this was like one of the rare movies where we saw a rupturing event, and that too, these are myotubes from patients which have muscular dystrophy. So otherwise, eukaryotic cells can take, and that was a huge amount of stretching. You could see that stretching of the cells.
Mononucleate?
No, multinucleate. There are multiple nucleates. In fact, this is not multinucleate, but this is a myotube which has basically now trying to, so it is A, I mean it's after a good number of days. So usually we can micropattern and have a huge rectangle kind of thing. With days the adhesion becomes weaker and it just pulls it, and most of the nucleides, and you have this scale deformation. Yeah. So yeah, I think that's the presentation. So you can ask questions if they are about it. Yes, no. Yeah, I think all questions have been asked. Okay.
So then instead of wanting to stretch single cells, you want to stretch whole sheet of cells.
Monolayer of cells is not problem. As well as I don't know. I don't think it should be a problem because all your cells are sticking to the surface. So each one. There are examples where people have grown a monolayer between two rods. And both the surface is free and then can grow a monolayer where each cell is adhering with non-glucose chromatins.
Is this di or diol that you're using?
No, this is actually calcium.
Okay.
Yeah. It is non-fluorescent outside the cell. It is permeable to membrane in that state, in the calcium state. It can diffuse to the lipid bilayer, go inside the membrane, get hybridized to calcium and break apart. Calcium becomes fluorescent and it becomes non-permeable. It cannot come out. And it can come out only in case of rupture. If you have membrane rupture, you lose fluorescence. In case you have repair and you have calcium outside, in fact, you can also see that. You have the intensity drop, and then in time you see that it starts to come up again, which means the membrane has been repaired. So that's why